Good evening. Welcome, everyone. On behalf of Fred Hutch, I'd like to welcome you to tonight's Science for Life presentation. Tonight's presentation will provide a glimpse into the exciting science conducted at the Hutch. Science for Life breaks down the concepts, skips the homework, and offers a chance for you to interact with world-class researchers in a fun and really informal atmosphere. We're really glad you're here. There are a few details before I introduce the speakers. There will be a dedicated time at the end of their individual presentations for Q&A, so they'll come up and sit and you can prepare your questions at that time. And to avoid interfering with the AV, if you could make sure your cell phones are turned off on airplane mode or silenced. Thank you. I'm really excited to be here tonight to introduce this group of people. They do work that is personally and professionally incredibly inspiring. Over the past decade, the HIV Vaccine Trials Network at Fred Hutch, or as we call it, the HVTN, has aimed to improve the process of designing, implementing, and analyzing preventive HIV vaccine trials. Several major achievements included establishing a laboratory program with standardized assays and systems allowing for reliable assessments across trials, setting statistical standards for the field, and actively engaging with over 60 site communities in 11 countries around the world. These achievements have allowed the HVTN to conduct over 80 clinical trials and make numerous scientific contributions to the field. They are the world's largest publicly funded network conducting trials to find a preventive HIV vaccine. Tonight's presentation will start by introducing the science behind HIV vaccine research, then look at the global effort to engage communities. We'll hear about one effort to develop new engagement tools using community-based participatory research methods. Once participants are enrolled in a trial, we'll be hearing about what actually happens in the laboratory to analyze the samples that are collected. And finally, we'll hear the perspective from our own local Seattle Research Clinic as they work to enroll participants and conduct HVTN studies. I'm pleased to introduce you to this evening's expert panel who are part of Fred Hutch's HIV Vaccine Trials Network. Each of them plays an important part in working toward a licensed preventive HIV vaccine and a future AIDS-free generation. Tonight's panel includes Gail Broder, Senior Community Engagement Project Manager, Jim Maynard, Director of the HVTN Communications and Community Engagement, Dr. Michelle Andrasic, Behavioral Scientist for HVTN, Dr. Maria Lemos, Fred Hutch Scientist, and Dr. Janine Manza, Medical Director for the Fred Hutch UW Vaccine Trials Unit. You stumped me. Thank you, Janine. She's usually so quiet. She's the one that pops up with a hello. So please join me at this time in welcoming our very first speaker, Gail Broder, to the podium.
And Rhonda, you forgot to introduce yourself. That was Rhonda Curry, our Director of Communications. So I get the pleasure of introducing you to some of the science behind our studies. My colleagues know that this is sort of my wheelhouse. I love doing this. One of my functions in the network is to be sort of a translator, and I always joke that I have to turn science into human. Because most of us don't go around speaking in 7-syllable words every day. So, and I tease our director all the time, did you really have to say immunogenicity? Because immune response means exactly the same thing. So we're going to try to bring it down to a little more human plane for all of us tonight. So one of the metaphors that I like to use actually is that you can think of your immune system like a tree. And it has some major parts. The adaptive, the innate systems. It has some pieces within the leaves, the twigs, maybe some fruit. And you don't necessarily have to know what each individual little bit of that tree is to appreciate how it all works together and sort of the sum is greater than the parts, if you will. So how do vaccines work? Their main function is to teach our bodies how to recognize and fight off invaders. I love this particular metaphor because every culture around the world that we've ever encountered has firemen. Firemen are global. So we want to show the body the virus, teach it how to recognize the virus, Our body's immune system sounds an alarm, calls our fighter cells into action, and then our goal is to ultimately put out the fire, right? So either prevent HIV infection from occurring or control an infection if it does occur. So what does a vaccine need to do in order to do that, to put out the fire? The first thing it needs to do is activate antibodies. And antibodies serve a couple of different purposes. One of them is to bind on, and in binding, they cover up some of the key components that a virus would use to attach to our cells and cause an infection. And so as you can kind of see in the lower right there, once those surfaces are covered over, that virus isn't able to interact with our cells and attach. That act of binding can also mean that the virus is neutralized, that it is prevented from being able to cause an infection. Binding and neutralizing have other functions as well, but that's the gist of it. And you can see what it looks like in this little animation. That's you in purple down at the bottom. You're the host, and your cells have receptors. You can think of them as little cup-shaped bits on the surface of your cells. And antibodies are these Y-shaped proteins. They float around. And if HIV were to float along, if you were ever exposed, the idea is that antibodies would bind onto that HIV and block its proteins on the surface from being able to attach to your cells and cause an infection. And I'm going to let that repeat and play through one more time so you can take it in. Our colleague Lisa Donahue made this animation. We're especially proud of it. So HIV, you can see in this case, we changed the color to indicate that it had been neutralized. So the next thing that we need vaccines to do is to boost up our T cells. And our T cells are the things that ordinarily fight back once an infection has happened. They're only activated once an infection has already taken place. And their usual job is to sort of be the sweepers. They go through and identify cells that look infected, are somehow misshapen, malformed. This could apply to cancerous cells. It could apply to any sort of infection that you might have. And T cells come along and do the sweep-up duty. So in particular, we're interested in the CTLs, which is shorthand for cytotoxic T lymphocytes. Try saying it 5 times fast, I promise. So CTLs is much preferable. And CTLs, their job is to be the killers. They are able to bind on, use the chemical properties within themselves to inject chemicals into a virus or a bacteria and kill that cell. And when that happens, the cell kind of self-destructs. The scientific name for that is apoptotic death, which is one of my favorite Scrabble words. If you ever have 2 O's, it comes in very handy. But it's basically cellular suicide. Because of having those chemicals from the CTL injected into the virus, that infected or abnormal cell can be cleared from the body. And how does this T cell response work? So it's literally like the defense system of the guy on the wall running surveillance with his binoculars. Our T cells are doing immunosurveillance all the time. They're looking for infection. They're looking for abnormalities. They're destroying those cells that don't look like they're shaped properly or functioning properly. And now you know why it makes sense that there's an HIV research program at a cancer center, because all of these diseases are things where our immune systems aren't able to fight back sufficiently to clear the disease from our bodies. So anything that our bench scientists learn that informs what we understand about the immune system is going to inform cancer work just as it's going to inform HIV work. So what's the time frame for these responses? If you're exposed to HIV, it stays localized in our mucosal tissue, so the skin of the mouth, the rectum, the vagina, the penis. It stays in those areas for the first 1 to 3 days. Then it starts to spread through our lymph nodes and lymph system and into those lymph centers within 3 to 5 days. And ultimately, it spreads throughout our entire bodies in less than 10 days. So our goal with the vaccine is to disrupt. We want to break up that cycle. And if we could teach our bodies to respond more quickly, to produce those antibodies, to produce those T cells, and disrupt that cycle, that would be the goal. So traditionally, and we encounter this all the time, the two most common ways of sort of old school technology to make vaccines was live attenuated or whole virus inactivated vaccines. Live attenuated meaning that it's the real germ, it's still living, but it's been weakened and weakened and weakened and weakened and weakened to just a microscopic percentage of the original. And the concern, obviously, is that with a virus like HIV, where we don't have yet a cure, although colleagues in the room here are working on that, I've seen them, we are concerned about the risk of potentially exposing someone to HIV if the virus hadn't been weakened sufficiently. Whole virus inactivated is what our principal investigator likes to refer to as pickling it. Using chemicals, using radiation, using heat to somehow kill that virus so that the whole virus is present, but it's no longer living and no longer capable of causing an infection. So, but like I said, these are sort of old school technologies. The most common vaccines that probably most folks in this room are aware of that use these technologies are the Salk and Sabin polio vaccines. But what we've seen in HIV so far is that in animal studies, they've been hard to manufacture in large scale, so to deliver them in a public health setting would be challenging. And in animal studies, there have been studies where the animals actually were able to develop the animal versions of immunodeficiency viruses. So, not great omens. But thankfully, we have new technologies. And don't worry about taking in a lot of words and a lot of details because they're too teeny and there's lots of it. But what I want you to know is these are all the new school ways to make vaccines. And at the HVTN, with one exception, we've tested all of them. We have not yet used a bacterial vector. But so using other kinds of viruses like some of the bird versions of pox viruses, canary pox, for example. We've used DNA. We've used other kinds of viruses from other animals. One of my favorites was trying to explain Venezuelan equine encephalitis in a consent form. It only infects horses. Don't worry, it'll be fine. So we have not been using the whole inactivated, and I'll explain why that's a dotted line in a moment. And we have not been using live attenuated, but we have been using all of these other technologies with the exception of bacteria. That hasn't come along to our network so far. The reason that I've put a dotted line on whole inactivated is that there is one Canadian research group that has now launched a clinical trial using a product made by a South Korean biotech company that is using a whole inactivated virus. It has been inactivated using both radiation and chemicals, so double inactivated. They did an initial phase 1 study in 40 people, and apparently the results were favorable because they've now been sending out press releases that they're going to launch a phase 2 study, but we haven't really heard any of the details yet. And part of our frustration is that they haven't presented any of this in vaccine scientific meetings. So none of us really have any of the details or know any of the scoop. We just know what's in the press releases. So it's a little challenging because we're not quite sure what to say about it, but we know it's happening. So for the moment, we're leaving it as a dotted line. But in our network, we're not using that strategy at this time. So why is HIV so challenging? The challenge is that we don't have any example in the globe of a person who has been able to naturally clear an HIV infection. No one has ever recovered from HIV naturally. So we're not able to look at a person's immune response to say, aha, that's what you have to be able to successfully clear HIV from the body. So we are truly doing that needle in a haystack that we've all heard about for years. The only clue that we have is this group of people that seem to have a natural immunity, and they're people who are primarily Caucasian of Western European ancestry, largely from areas like the British Isles, France, Germany, that area, areas that had bubonic plague in the 12 to 1500s-ish. And there seems to be a genetic mutation that has come out of that region and the experience of the bubonic plague. And they have a mutation to one of the receptors on their T cells, those little cup shapes. And so if you can imagine having that misshapen receptor, HIV is now the key trying to fit in a broken lock. And it doesn't attach, it doesn't fit. So that gives us some clues. Would it be possible to induce that kind of a genetic mutation in a healthy population? Something to consider. So we're now in a position of having to sort of one-up Mother Nature. We've got to do one better and find an unnatural protective immunity because we don't have it as we're born. And of course, the challenge is that this is a response that has to be in all the right places at exactly the right time and ready to be deployed rapidly because HIV gets into our whole body in less than 10 days. So whatever that immune response is, it's got to be ready to go and it's got to be ready to react. Who participates? So we are doing in our trials just adults, typically 18 to 50. There's sometimes a little variation. You might see a trial that only goes to 40, a trial that only goes to 35. We can talk more about that later in the Q&A if you're interested. But most importantly is the second half of that phrase, and that's that people understand the risks and benefits. We're not selling used cars. When we're doing research, we want to make sure people understand what they're getting into, what risks there may be, what risks we may not even know about yet because some of these products are new and novel and being used in the first time in humans sometimes. So we spend a lot of time on informed consent, and I'm sure Janine can tell you more about that from her experience at the site later if you're interested. But the upper age limit is kind of an interesting thing. Our immune systems tend to change and evolve as we age, and not necessarily in helpful ways. They tend to sort of diminish as we age, which is why you may be noticing this winter there's been a big push for pneumonia vaccine commercials. I've seen a lot of that. There's now a pneumonia vaccine available for anyone over 50. If you haven't had one yet, might be something to talk about. There's a new vaccine for, I think it is also for over 50 against shingles. If you had chickenpox in childhood or younger adulthood, that virus is still alive in your body and can remanifest as shingles at a later age. So shingles is now a vaccine also for older adults. So we want to try to avoid those challenges of a diminishing immune system and work in a, if you will, a more pristine environment. So we're trying to really enroll people who are younger because their immune systems are still the most robust. And at least currently epidemiological data says that it is primarily younger people who are being infected with HIV. So we know that whatever vaccine we ultimately find, it's going to have to work in younger people. And so that provides the impetus to really enroll those younger people in our trials. Diversity is also really important because with this disease, we still don't know yet if it matters. Does it matter what gender you are? Does it matter what your racial or ethnic history is? Does it matter what part of the world you live in where there may be different nutritional issues? There may be different pollutants in your environment. We just don't know yet. So the burden is really on us to try to include as wide a swath of humanity in our trials as we can. And so Rhonda mentioned that we have now over 60 sites in 11 countries, and that's part of our rationale. We want to make sure that if we find a vaccine, that we can say it's going to work in Peru, it's going to work in Mozambique, it's going to work in South Africa, it's going to work in San Francisco, it's going to work in Switzerland. We want to be able to find something that's really going to have global impact. Some of the common misconceptions that we bump up against, probably the most common is that people think a vaccine already exists. And part of the challenge with that is that we don't have good ways to talk about potential vaccines. So when you hear about vaccine research, it's the same word, vaccine. So how are you supposed to know if it's a licensed one or an experimental one? It's hard to say. So this is a really common challenge that we bump up against. I think even more so the concern that a vaccine, it could actually cause you to become infected with HIV. And so why it is that people seem to remember from junior high biology class about whole virus inactivated and so on. Why do we remember that those are the two ways that vaccines used to be made? That seems to be a little fact that we retain for years and years and years. So we have to spend a lot of time explaining the fact that there are newer technologies and genetic engineering methods to make vaccines in other ways that are safer and don't have to use real HIV. And last but not least, we encounter often that people misunderstand that you might have to be HIV infected to be in our trials. And in fact, it's exactly the opposite. Vaccines are designed to keep you healthy. So we want to enroll people who are uninfected because our goal is to keep you that way. But because particularly in the media, we tend to only hear about HIV when we're talking about infection rates around the world, there's a bit of a misrepresentation, I think, in what people hear in public circles that contributes to that misunderstanding. So we spend a lot of time on these three questions. I'm looking to some of my colleagues in the room, James, Kim. Do we spend a lot of time talking about these three things? Yeah, James is going, yeah. So they come up again. I've been doing this work now for 14 years. I think I've probably answered these three questions in every single presentation I've ever given. It comes up all the time. So I'm going to stop there and turn it over. Oh, no, I'm not either. I have one more slide that I forgot about. So this is actually what we're all about, is trying to figure out this puzzle about how do we prevent HIV. And we often say that a vaccine is the best long-term hope because vaccines historically, vaccines and fresh water have been the two things that have stopped more infectious diseases than anything else in all of history. But because we don't have a vaccine yet, things that work in the short term are just as important and just as vital to curbing this epidemic. So we really work hand in hand with our colleagues to look at ways to prevent mother-to-child transmission, ways to roll out male circumcision programs in cultures where that's acceptable, using antiretroviral treatment as prevention. One of those ways is, you may have heard about PrEP, which is an acronym for pre-exposure prophylaxis. There's currently one medication that's been approved for that purpose. It's a drug called Truvada that contains actually two medications. They're the same kinds of antiretroviral medications taken by people who are living with HIV, but they can be taken preventively, a pill a day to prevent HIV. It's over 90% effective if taken daily. So you can sort of think of it like birth control pills, but for HIV. Same idea. So we see ourselves as part of this bigger picture. We have colleagues working in microbicide research. We have our group working on vaccines. If we have time tonight, maybe in the Q&A, we can tell you a little bit about antibody-mediated prevention, which is the newest thing we're working on. But really, it's about all of these things working together and creating kind of that quilt that hopefully by offering more opportunities, more options, that in the same way that the family planning movement has learned that if you give a woman more options for birth control, you're more likely to find one that she'll actually use. Same idea here. We want to have as many tools in the toolbox as we can find because they're going to serve a variety of people's needs at different points in times in their life for different lengths of time. You may use one in short term, one in the long term, but we see ourselves as part of that big picture. And now, I turn it over to my manager, Jim.
Just good evening and welcome. I know that you're giving up an evening to be here. I want to test really quickly and make sure that I'm not hallucinating. So bear with me for a minute. Who here has noticed March has been a wee bit dark and wet? I saw in the news that there was only something like 2 truly sunny days this month. Thank God we're all so much in love with Seattle. But I swear to God, I saw a sunbeam as I was walking in tonight. Did anyone else see a sunbeam? Yay, I'm not crazy. Then I will go ahead and I'll do the presentation since I'm not crazy. That's wonderful. What do we need? We need a safe and effective HIV vaccine because that really is the gold standard in disease prevention. Vaccines have almost totally eliminated things like measles and mumps. Smallpox is totally gone. Polio is almost totally eliminated. That's what we need. And one of the best kept secrets in this town is that the Fred Hutch is ground zero in HIV vaccine development. Who here knew that this evening before you came? I mentioned it to my friends. I say I do HIV vaccine work at the Hutch, and they always give me the eye saying, the Hutch? Why on earth would you be there at the Hutch? You know, it's because we have world-class scientists here, number one. We have an awful lot of skill and talent here. But it takes a village to actually get to a vaccine. One of our principal investigators, Glenda Gray, says, to be successful, we need more than just clever scientists. So if you are a clever scientist, my apologies this evening. But there's lots of other people. There's people that work in data, in pharma, in lab. There's lots of people that work at the sites, and you'll hear some of these people speaking today. But you know, they're actually not the heroes. There's one class, one group of people that are actually more important, I believe, than the scientists, more important than anyone else that we encounter. It's our volunteers. Volunteers are the absolute heroes of this or any vaccine development. They give the ultimate gift. I always imagine, would I be willing to do that? To give up what is more personal than your body and your own immune system? To allow us to attempt to alter your immune system. They're making this beautiful gift to us. And we recognize that, and we realize that nothing happens without the volunteers, simple and pure. So how many volunteers, how many heroes do we need? I love the graphics. I didn't do that. Someone did it for me. Superman flying in. So let's just stop and think for a minute, because I'm the one who, when the rubber meets the road, will be responsible for the volunteers. I'm the director of community engagement. So if you see some bags under my eyes tonight, you'll know why. Let's start off with, and I'm just going to go through our efficacy trials. An efficacy trial is a very large trial that enrolls many, many people into it. And that way you can get not only more safety data and some immunological data, but also find out if it actually works. I mean, half the volunteers will actually get whatever we're testing, the other half will get a placebo, and then we look to see infection rates, and then we'll be able to tell whether it's working or not. Okay, what do we need to do here? HVTN 702 is going to be testing with men and women at risk in sub-Saharan Africa. And believe it or not, we need 5,400 volunteers. 4 to 5 years worth of hard work before we find out whether the vaccine is efficacious, whether it actually works. That would be fine, except they added another one. They added HVTN 703, which is a whole different prevention modality. It's testing it in women in sub-Saharan Africa, and that's 1,500. So now I'm up to thinking about 6,900, and I'm not even good at numbers. 6,900 participants we'll need to find. We could end there, except that we're also enrolling HVTN 704, a sister trial to 703. We're testing that in the Americas and in one site in Europe. We're testing that in men who have sex with men and transgendered individuals, and we need 2,700 volunteers to make that work. I'm going to finish up with HVTN 705. Women in sub-Saharan Africa, we need to enroll 2,600 participants. So that brings the grand total, and just think we're starting from the Fred Hutch, of 12,200 volunteers. I'm going to throw out one really quick phrase that I didn't throw out before, that it all happens at the Hutch. Things begin here at the Hutch, and it's happening in your backyard. So someday, and I can say this because I'm not a scientist, someday when we do get to a safe and effective vaccine that would be licensable, you'll know that it happened in your own backyard. But we need more than just the people at the Hutch to make it happen. We need those volunteers. We have sites. Actually, the exact count is 62 sites in 11 different countries. So we have 62 sites, and at each of those sites there's very talented staff. And their job is to actually conduct the trial, and the HVTN helps them in that process. And one of the things they do is to actually find these 12,200 participants with some very talented staff. Here we have one of the heroes, and this is at our Nashville site. And you'll see leaning over his shoulder Vic, who is a CER, That means community engagement and recruitment person. So he's a specialist, and his job is to walk out into the communities and not to simply parachute in. The old model for doing any kind of research for scientists was 100 years ago, you kind of parachute down into a community, right? You find the people you want to do the research, you grab all of your data like a greedy little scientist, and then you just disappear with it, and that's it. We cannot do research like that because these are real communities, these are real people, these are brothers and sisters. So this is another group. This is actually the Seattle staff. So San Francisco, excuse me. So you see the principal investigator and you see people in the outreach staff. And they are experts in going out into their community, connecting with people, building bridges. And the name of their program is actually called HIV Bridge. And that's exactly what they do, bridging to the community to help find these life-saving interventions. Now, we like to say that the VTN does not work in communities. We work with communities as full partners in the research process. And if someone is your full partner, you don't bring them in at the very end of a deal, do you? You bring them in at the start of the deal. So we have community members in all 62 of these communities. This is something we work on very hard, well ahead of time, with something called community advisory boards. And when we're writing the protocol, I like to think of the protocol almost like the U.S. Constitution, because once you write it, it's there. And we have to follow it closer than it appears people are following the U.S. Constitution these days. We have to follow it word for word, and community members have a strong word in the room. As the scientists and the other specialists in the room are talking about what we'd have to do in the community members can actually put a stop and say, no, I don't think you can ask people to come in that often. I don't think you can ask them for a rectal biopsy every other time they come in. I don't think you can do this, or yeah, that kind of sets me on edge when I think of it too. There's lots of procedures I wouldn't want. They're also involved in every single board and committee of the HVTN at the Fred Hutch, including our governing body, which is the Scientific Governance Committee. The volunteers help run the show. We don't run the volunteers. We begin by knowing that an educated community is an empowered community. So what the volunteers get is they get our highly trained staff at all 62 locations, and they go and they build bridges to the community by getting out into the community. You're not going to get 12,200 volunteers by sitting on your duff in a research lab or one of the CTUs and waiting for people to come to you. You have to go to them. You might go and do an information table, doing outreach as part of community education. You might be teaching a class at the local college or a high school. You might be doing a radio show. You might be at an AIDS walk or an AIDS ride, talking to people as they're coming and going. There's all different events that you're a part of, because to do this, you need to get out into the community. You need to become a part of that community you're addressing. What do we do here at the Hutch? I want to get back to the Hutch a little bit. We also have very talented staff that are part of the Hutch staff. I head up a communications team and what's called a community engagements team that Gail heads up. So between those two, one of the things they do is come up with actual tools, because if we're sending people out into the community, we want to make sure they have a full tool belt in order to actually do the community education and hopefully eventually recruitment. And I know I only have 10 minutes, so I'm going to fly through these if you don't mind. Study brochures, postcards, flyers and posters, palm cards, pull-up banners, flip charts, community education slides. All of these are culturally competent. As we develop them, we actually involve CAB and community members in developing them to make sure they're actually useful tools that they can use out in the field. We also do study promotional print ads. So we need to get the good word out there that we're doing a trial in whatever community. And our educators and recruiters can only get out and see so many groups. So something, again, we do centrally at the Hutch with our communications is we produce advertisements to help get the word out. We do things like transit ads, so people can be riding on a bus and see, help stop HIV. Go to this website, call this phone number. We have indoor advertising. You might be in a gay bar on a Friday night. You might go into the men's room and there's a poster there waiting for you. that you can read talking about what's going on and how you can become involved. Banners, magazines, newspapers, all the different ways, event program guides. I used to actually be a recruiter way back in, I think it was dinosaurs back when I was doing it in Boston. And back in those days, if I wanted to advertise, what I did was I called up a newspaper and I took out an ad, and they placed that for me. We really weren't doing a whole lot with this new thing that was called the internet. So it appears that the internet is a very good way to recruit, and that's what we've discovered down through the years. In some of our recent trials, 1/3 of all people that are recruited by using the internet. Not all the good ideas come out of the Hutch, by the way. So I want to throw up something that San Francisco did. I love this image. I think this is powerful, to be a warrior fighting HIV. And that's something they push out online. Everything is geared towards the cohort. The cohort is the name of the people you're recruiting. Who can guess what my cohort is? This is for the United States, and this is actually advertisements that we developed here centrally at the Hutch in Communications to recruit men who have sex with men, what you might think of as gay and bisexual men, and also transgendered men and transgendered women. We do lots of research that is data-driven, and we place these many ads on internet sites. It might be something as innocuous as just putting them on Facebook. It might be something more targeted, like going to Manhunt or Adam for Adam, which are gay social sites. Another real cutting edge for us, and that's what you see, these two flashing images, which I love, by the way. Nowadays, if you're a gay or a bisexual man, especially, well, I guess anywhere in the world, there are actually handheld apps on your smartphone. On your smartphone, you can have something called Scruff or Growler or Grindr. Who here has ever heard of Scruff or Growler or Grindr before? I'm not going to say who has it on their phone. It's a dating app, and it's a wonderful one. So ask someone who raised their hand if they'd be willing to show it to you if they happen to have it on their phone. It's actually an app that will show you how many feet away an other member is standing with their picture and lots of different data on that person. So that way you can set up a date. This is a very powerful tool for getting the word out, because we'll find volunteers everywhere. But again, the onus is on us. We need to do the hard work to find the people that are willing to put everything on the line for us, again, to offer their immune system for us to work with. Anytime they click on something, they end up going to a study website. So if you clicked on Grindr, what are you looking at? You're looking at an ad that might look somewhat familiar to you if you're part of the gay community. It appears to be two men who appear to be getting very close to each other. So when you go to the site, at these recruitment sites, you can find out detailed information about the trial itself. You can find out what would be involved being a volunteer, what kind of time is involved, and maybe some of the procedures. Not everything, but some of the basics to be a volunteer. You'll also find out where the study clinics actually are. And if you're interested, and also we do them in multiple languages. For the sites that we're doing in Africa right now, I believe we're up to how many? 9 languages. So people can click on their language, so they don't have to read English, and they can find out about the trials. And then if you're interested, you can fill out an actual form that says, yes, I'm interested, here's my name, email address, phone number, a good time to call me, have someone contact me. And when they hit send, that will go to one of the 62 sites immediately, as well as it goes to us. And that way people will know that they're going to get a phone call or an email, some sort of contact to find out about the trial. But when it comes right down to it, community education and recruitment is not about advertisements. It's not about Scruff or Grindr. It's about people to people, nose to nose. People are recruited one at a time because it's an individual decision to be involved in a trial. So I like to refer to this as feet on the street. These are the people that get out on the streets, again, the people I've been referring to as the CERs. And they get out there. This young guy was in church preaching, which I think was a great idea. These two individuals are part of the San Francisco team. Their whole team dresses up as superheroes. Do you love that? They dress up as superheroes, and they're just out on the streets in San Francisco. And I'm told people don't bat an eye when they see them on those streets. But they're out walking up to people saying, would you like to know about a way that you can help stop HIV once and for good? They might be in a bar in Atlanta or in Dallas or in New York City or in Cleveland or right here in Seattle, where we also have a wonderful clinical trial site. I love these guys. This is from our Lima site, one of the Lima sites, and they have branded their own superheroes, and they have the name of the clinic. So what they'll do is they'll go out to like a huge gay discotheque on a Friday or Saturday night where there may be 1,000 men, and they simply show up and they'll be on stage and they'll start to perform, and then all the men become very interested in them performing, and that gives them an opportunity then to say something about the trials and the ways people could become involved. And lots of other opportunities to be feet on the street. Last but not least, this is just for your curiosity, for your interest. When I used to be a recruiter myself, it meant a lot if I had a table at a bar, if I was out at some big event, if I was talking to people on the streets, to be able to offer them a gift, to say thank you for taking the time to listen to me. And here's just a few of the couple dozen different things that we do. We do this locally, again, at the Fred Hutch in our communications, and we send them out directly to the sites. I was given 10 minutes, of which I think I just took 15, so I will just simply finish up again with saying that if you take one thing away from this night, I hope you take a sense of pride that as Seattleites, this is happening in our backyard. And if there does come one day, and I believe there will be, a safe and effective vaccine, it's going to become because of us, and it's going to happen at the Hutch. Thank you.
Hello, everyone. So I'm going to talk a little bit about some of the social and behavioral science efforts that we engage in around community engagement. This is a picture of one of our community consultations in sub-Saharan Africa. My colleague Linda is in the sweater there. And this was a consultation in an effort to create the videos that I'm going to talk about today. So one of the things that Jim and Gail talked about is the fact that all of us are very engaged at the community level. So as the lead social and behavioral scientist at the HVTN, often community members are talking to me about some of the challenges that they see in their communities, communicating different scientific concepts or issues that come up around vaccines or prevention generally. So often in these conversations, we're able to identify a need that the sites have. And so one of the needs that we found as we were embarking on our AMP trial, and AMP is the trial testing broadly neutralizing antibodies, which was a it was a new approach for the VTN. Prior to the AMP trial, we had been testing vaccine products, which required injections. Broadly neutralizing antibodies required infusions. So, talking to people about the difference between injections and infusions in that process, the sites were somewhat concerned about how that would play out and how they could effectively convey that information to communities who knew us as a vaccine trial network and our sites as vaccine trial units. So, as we identified this need, the folks of us at CORE decided, well, perhaps we should look into how we can assist the sites with some community engagement materials around some of these challenges that are being presented. So in multiple conversations with community stakeholders and site members, site PIs, CAB members, we identified that the best way forward would be to create a series of videos to explain broadly neutralizing antibodies and to explain some of the concepts and what a participant might expect when they go to the site and so forth. And one of the other great things about the VTN is we have several junior investigator programs or scholar programs. So through the RAMP Scholar Program, which is a program that focuses on bringing medical students, Black and Latinx medical students, specifically into the network for a short-term research project, either over a summer or a year, to get medical students interested in doing vaccine research. So one of our RAMP scholars who was in San Francisco for a summer had already done a community-based project looking at conveying information through the use of videos. So we utilized that approach that we had this sort of preliminary data for to move forward. And in all of our work, in my unit, as we work on community engagement, we use a community-based participatory research approach. And the main tenets of that approach is that the community, the stakeholders, are the experts, and they are involved from the very beginning, from the very moment that we have an idea. Community members are involved, and we identify ways to involve them to ensure that we're getting their feedback, guidance, and direction throughout the entire process. So as we started this process, we worked with community members to identify where along the line of creating these community educational videos we could get feedback. And so we identified several stages, and we worked with our video company as well, because this was my first video project. And many of our community members, it was their first video project. So our video company worked with us to help us identify where along the production of these videos we could get feedback. And we identified these stages in the work. So script development, basically we worked as a team to develop scripts and then disseminated those out to get feedback. We got feedback once those scripts were turned into screenplays, and then once those screenplays were turned into animatics, which was a rough sketch of the animation concept, because early on we had decided also as a team that we were going to do animated videos, and then voiceovers whenever we had several people audition for the voiceovers, and then sent those out to the sites as well, music, final review. So those were the stages, and at each one of those stages, we identified how we would get community input. So as I started saying before, we had a project team that included site staff, scientists, community members, CER members, who drafted scripts for a set of 4 videos. And the 4 videos came from all of our discussions with the stakeholders that I mentioned before. And from these scripts, we held a series of 7 webinars where we went through a line-by-line review of the script. And anyone who wanted to attend the webinars could attend the webinars, and we worked with our sites to ensure that we had community stakeholders, CAB members, site staff on those webinars so that we could go through line by line and talk about what was appropriate language, how to make the concepts more accessible to everyone. And then we utilized the feedback that we collated from those 7 webinars to provide that to the animation company. So this is an example of the script that we used. And this, I think, is probably it does not look like it's the final draft. So it's probably like 2 or 3 drafts down the line of the final script. But this is what people were looking at when we were on our webinars, going through the line by line and deciding what we would include in the script. So stage 2 then was the screenplays. And so the animation company basically gave us shots of, or this is what the screenplay looked like. So on one side, it would describe what the visuals would look like on the screen, and the other side would have the associated text with those visuals. And so we sent those storyboards out to everyone who was on the webinars. And so we, after we had those initial webinars, we made sure that we had everyone's contact information. So we sent out the screenplays and allowed 2 weeks for feedback. And I think the critical point to make with allowing for 2 weeks for feedback is that you remind people a week into it, and then you remind them a week and a half into it, and then a day before you want the feedback, that, you know, we're looking for their feedback. And that really helped facilitate getting a large amount of feedback on these screenplays. And then again, the suggested revisions that we received, we collated as a team and then sent on to the animation company. And then they turned those screenplays into animatics. And we took the animatics on the road, if you will. We basically worked with our sites to develop regional meetings where we brought site staff, CAB members, community stakeholders in to show them the animatics, the rough drafts of these animated videos. And this stage was critical, and it taught us a valuable lesson, because what you think you want is not always what you get, because people have an idea of what they're going to see, and then they see something and it's not quite the idea that they had. So this was the stage where we received the most useful critical feedback on these videos. And it was incredibly important to ensure that all stakeholders were engaged. So not only did we have the West Coast and the East Coast consultations, but we worked with Stefan Wallace, who I know is in the room somewhere, who is you know, a leader in the house ball community, and he facilitated you know, us having an hour at the House Ball Summit in LA. So we got to show the videos to some of the leaders in the house ball community across the nation. Does everybody know what the house ball community is? So the house ball community, and I hope I do this justice, Stefan, is basically an organization of houses of that largely are comprised of Black and Latinx and other people of color who identify as gay, lesbian, bisexual, some are cisgender, but they basically formed these houses as families, you know, some surrogate families, but others, you know, just families to help combat stigma against homophobia or homosexuality, homophobia, racism, and so forth. And these houses often compete in balls where there are several categories. I was at one the other night. It did not start until 12:30 and went until 7. I did not make it that long. But anyway, they are, you know, I think a really great resource for largely Black and Latinx, lesbian, gay, bisexual, transgender individuals, and create a social network and social support system that is critical, I think, for communities that are often marginalized. And these communities also happen to be the hardest hit communities with regard to HIV. So it is critical that we make community engagement materials that speak to these communities. So having an opportunity to present these materials to the houseball community was incredible. And besides that, we also presented at a Boston stakeholders meeting. So we had the opportunity to present these videos in several key arenas, and again, compile the feedback and give them to the video company. So this is an example of an animatic. You can see it's just a very rough draft. It's almost like scribbles on a whiteboard. Which people had a big problem with, because they're like, this is elementary, and this is, what, are you paying money for this? So just getting through the, the idea that this was a rough draft of what, like, that was, I think, a really challenging hurdle to get over. And actually, as we create videos now for Sub-Saharan Africa, we're thinking through how we might avoid the animatics issue. So stages 4 and 5 were music and voiceovers. And for the music, we also had the good fortune of getting Damon Humes, who is a legend, an icon from the House of Blahnik in the houseball community. And he did the music for us and gave us several samples of music. We gave that back to the community to get the input on the music, what kind of music we should have for the videos. We also got several samples of voices that would be talking during the animation. And again, we sent those out to everyone, gave them 2 weeks to get back to us, and then sent that to the company. And then the final stage was pulling all of this together and getting everyone's opinion. And this was critical. Once we had the final videos, we really had to work very hard to make sure that everyone was giving us feedback. Because, as you might imagine, for cost effectiveness, we couldn't take the final videos out again. So, we had our chance to take them into the community, and now we needed to do whatever we could on the ground to get feedback for these videos. This is another picture in Mozambique, the Mozambique staff, I think, yes. And so the lessons that we learned in creating these videos is that, with all of our community engagement efforts, we need to make the effort up front to ensure that we have community input, feedback, and guidance at every critical point of the development of these community education videos, and that we really need to adhere to a CBPR approach, like a true CBPR approach, in order to ensure that our final product is something that is useful and utilized by the sites and the communities. And again, allowing enough time for review and making sure that during the review process, you're prodding people along. Hey, just wanted you to know we only have another week for the review, and then sending it out again to make sure that it's right in the email that you're giving them the reminder for, so they don't have to go and look for it. And then we have these 4 amazing videos that we created for the AMP project. And I'm happy to say that I talk to everyone about these all the time. They seem to be being utilized not only here in the United States, but in all of our sub-Saharan African countries, in Peru, in Brazil, and soon in Switzerland. We are translating them. We are in the process of translating them now to French for our Swiss site. So I just want to play one of them. This is the shorter one. If you're interested in seeing all of them and the process that we went through, you can go to ampstudy.org. So that's one of four. And I just want to say, my last thing is that these were so successful that we are now embarking on another video project, which has now become sort of a sub-unit of our unit, this production of videos, where we are making 6 videos that will be used in South Africa to combat some of the misinformation and miseducation around HIV, HIV research, and vaccine research. So stay tuned for those. We hope to have them by the end of the year.
Well, thank you everyone for joining, and thank you to all of the earlier speakers for setting me up for telling you a little bit about what happens to the specimens that we are able to collect from these volunteers. And I have, as you have heard, there is no doubt that the hardest part of this research is done by the volunteers. I'm going to tell you about what we do in the lab, but I also want to highlight that we have many considerations about how to treat these specimens collected with integrity and respect to honor the will and the desires of the participants. So I guess I'll do this. There are many reasons why we collect these samples. Most of them are listed here, but mainly we want to know whether a vaccine product is safe to use in humans or an antibody-mediated protection antibody is safe to use in humans. We want to figure out what dose it would need to be used at. We want to know if it generates a strong immune response that we think could help. And we also want to know how much it varies among different individuals. Gail mentioned earlier why we wanted a very broad spectrum of people in order to test them, because we know every immune system is different. So we want to make sure that that a vaccine works in the largest number of people. Additionally, we want to know if the immune response, of course, protects against HIV. And normally, in our research, we need, of course, to answer the first questions first, and then move on to test whether it's totally effective. So in our protocols, we collect many different types of samples. We mainly collect blood in most protocols, but there are some and a subset of protocols where we collect specimens from other parts of the body. As you all probably know, the most common way of transmission of HIV is through sex, and therefore we want to know what happens at the surfaces where HIV can encounter the body. As Gail covered earlier, we believe that if the immune response is present at those early time points during infection, we have perhaps a better chance of preventing the transmission. So we really want to try to figure out what is happening in these sites of transmission. So these are some of the samples that we collect: cervical and vaginal secretions and biopsies from females, semen, rectal fluid and biopsies, but rectal fluid and biopsies for both men and women, stool. And in one protocol that is now complete, we have even been able to collect foreskin samples. In the top, I'm just showing you a blood sample and what it looks like. And if you're interested, in the bottom, you can see what a tiny biopsy looks like. Of course, in the big picture, it looks gigantic, and in reality, it's smaller than half a piece of rice, to give you some perspective. The lab side of things has many, many roles, and the first one is to make sure that we process all of the samples that are collected at the clinic. So in addition to all of these clinic sites that you have heard about, we have tiny labs next to each of those clinic sites so that every sample that gets collected gets processed appropriately. And processed appropriately means not only that we prepare it in a way that can be stored, but it also means that it gets blinded so that no one but the clinic has your personal information if you're a volunteer. So from then on, every sample will be associated with a number that cannot be necessarily traced back to that person except with permission from the IRB, which is the ethics review board of these protocols. So the second thing that happens is that we need to properly store it and transport it to the sites where the testing is going to happen. Here in Seattle, we have our own processing lab. We also have our own transport team, and we have our own storage team, and we also have a specimen testing team. That doesn't always happen in all of these other sites, so you can imagine that we are often receiving many, many shipments from all over the world to receive these samples. The samples close to clinical research sites will be arriving in many different ways and need to be processed in many different ways. On the picture on the right, what you actually see is blood that needed to be prepared to collect serum. Serum is the yellow part of that blood that most of the cells are in the red part, and then we are only interested in antibodies for this kind of assays, so we collect the yellow part and store it for measuring antibodies. And of course, sometimes we have samples that come already frozen or that need to be stored immediately. All of these have different temperature requirements, so we have to do a lot of babysitting of these guys. At the Hutch, we additionally have all of the quality control for many of these different clinical sites. So we make sure that everyone is processing exactly the same way so that all of the samples can be compared. We do our training or certification of all of these different sites. We also have a gigantic freezer farm where all of these samples, or many of these samples, are collected. We have others in different sites because we have to make sure that if anything were to happen to Seattle, there's still vaccine research that can continue. But certainly the Hutch is in charge of many of these samples. And we have to take care of them because we want to make sure that the vaccine that we tested 10 years ago can be compared with the vaccine that is being tested now, and that we have a way to do those kinds of comparisons. Sometimes the science evolves and we change our way of thinking about how a vaccine was working. So then we need to go and look back whether an older vaccine was doing something that we didn't expect at the time. We also have the duty of discarding any specimens from participants that did not give consent to the use of our specimens for other purposes. If any of you has ever seen a consent form, you will have all of the instructions of this specific trial, and then at the end you'll have a page where you would consent that if the samples have already been collected for this study, but then later on we find that it would be the ideal sample to use for another study, you can agree to that use. But some people do not want to agree to that, and then we have to find those samples 5 or 6 years later and discard them so that they are in agreement with the ethics board. In addition, we do all the laboratory testing. We do a lot of quality control for all of these samples and the way they are stored. We do specialized diagnostics. Some of the things that, for example, we need to be able to separate is what the immune response would look like from someone that has been infected from the immune response that would look like from someone that has been vaccinated, so that we can tell those differences. We also need to find, for example, HIV very, very early. So we have a specific HIV virology agreements so that we can find HIV much earlier than the majority of testing that occurs in a regular clinical site. And we have, as Gail covered, an interest in studying what's called the adaptive part of the immune system, which is the one that changes with vaccination. So the one that learns from having a vaccine in the body. And that contains both antibodies, which are in this picture in the red little dots that you see on, I guess, your left. And we're also interested in the cells that fight the immune system. Mainly, we have been interested in B cells, because those are the cells that produce the antibodies. And we have been interested in T cells, and there's two subsets of T cells, the CD4 T cells and the CD8 T cells. CD8 T cells are these CTLs that Gail covered earlier, and CD4 T cells are these very unique cells for HIV. On one hand, they are the target of infection, and then on the other hand, they are the officer that regulates both the B cells and the CD8 T cells. So we want to study all of these in the context of an HIV vaccine. Some of these specific assays that we run for many of our vaccines include understanding why or whether a vaccine is able to generate an HIV-specific response. We want to know what kinds of proteins is the B cell or T cell immune response producing. And in this picture, I'm showing you kind of a diagram that describes in a simplified manner kind of how we do this. So as you can see, we obtain the blood sample from the participant, and then we store it in a way that can be preserved. And then we essentially have a method called cryopreservation, which means the sample is frozen for a number of years. Then we have to kind of bring it back to life, and we use some stains and essentially we paint the cells of different colors to figure out what cells are present. And we pass one cell at a time through a laser to look at what colors it got. And as we can do that, we can draw essentially one dot in that blue diagram up there for every cell that goes through. And we can tell how many CD8 T cells and how many CD4 T cells were present in that blood sample. This allows us to figure out then out of these, we can figure out which ones are specific for HIV, and we can figure out the specific ones for HIV, what kind of proteins are they producing to fight a potential invader. In addition, we want to know what parts of HIV is the vaccine generating a response to. As you might know, we are not only interested in testing HIV in many different parts of the world because people are very different, we're also interested in studying the vaccine in many different parts of the world because the virus is very different in different parts of the world. So we need to make sure that the parts of HIV that we make a vaccine against are parts that are common to strains that circulate in Africa and strains that circulate in Asia and strains that circulate in the US. Lastly, we have abilities to tell whether the immune system is able to kill HIV-infected cells. So for this, we essentially, in the lab, infect some of the CD4 T cells that I told you are targets of infection. And then we culture them with CTLs, which are supposed to kill, and then we measure how many of those CD4s are able to be killed in our wells in the lab. And so we can have an idea of whether those CTLs are really good or whether they need a lot of virus before they can kill. We can ask questions about whether the immune system would prevent infection at locations of exposure. And the picture I'm showing you here is a picture of actually foreskin explants, but we do this with many of those biopsies. And we're very interested in these kinds of assays because what it allows us to do is to have a sample that comes from the region where HIV acts, and then we can culture it in the lab the little piece of tissue and then expose it to HIV and see if the immune response that has been placed normally at that area is able to prevent infection or not. So we're really excited of potentially having a way to have a surrogate way of testing exposure and whether protection can be mediated at these sites. Additionally, we're very interested in understanding relationships with the microbes that live in all of these different sites, because inflammation seems to matter in terms of whether you're at risk or not for infection. So we want to be able to figure out what microbes are present when an infection happens, and also we want to know if any of our products alter the kinds of bacteria and viruses that live in these sites. When we have finished all of our experiments and we put all of that data together, we send it to the statisticians who are going to receive data from very different labs and analyze it to figure out what the answer to our question is. And for that, SHARP is the Statistical Center for HIV, AIDS, and Prevention, and they collect a lot of this data and put it together here at the Hutch also to have the results of our trials. Thank you so much.
I'm just going to grab a water.
Okay. So I get really the pleasure of wrapping up the rest of this talk by talking about how the Seattle community is engaged in all of the research and efforts that you've been hearing about this evening. So I'm here representing the Seattle site, which is one of the 62 sites that my colleagues mentioned, and we are in the corner up there, the Seattle HIV Vaccine Trials Unit. And so what I'm going to describe in the last few minutes is how the information that you've been hearing about this evening plays out in the Seattle community and at our research clinic. So this is probably clear from the earlier presentations, but just to, again, show the connection, both the network and the unit are located in Seattle, and as you've been hearing, the network coordinates our research, provides scientific leadership, the central laboratories, data management, and statistical expertise. And what we do at the site is we're providing community education, recruiting, and enrolling volunteers. So what does that look like in Seattle? So we definitely take to heart the guidance that is shown here from UNAIDS and WHO guidance on HIV prevention trials, and involve communities, both engaging communities at the start of research, as well as keeping our communities apprised of information as it develops. And what you're seeing in this picture here is one of our laboratory scientists who's speaking at a HIV Vaccine Awareness Day, giving a scientific presentation on a day that also involved trivia and other contests and lab tours, and was an opportunity for community members to hear about the work going on in Seattle. On a more regular basis, our CAB, or Community Advisory Board, that I know Jim was talking about CABs around the world, we certainly have a CAB here in Seattle. Our group has been in existence since 1992. We have a very committed group of volunteers who come meet monthly, talk about HIV prevention research, HIV vaccine research, the work we're doing, and bring their own perspectives from the community and their own professional expertise in a number of different areas back to our group as well. And what you're seeing on this slide are just a few examples. The large group picture is actually a cross-CAB collaboration, because apart from our research clinic conducting vaccine research, a number of other HIV research units in Seattle, and they also have CABs, and the CABs will collaborate as shown here. This was an event that involved a workshop and visit to the race exhibit at Pacific Science Center. You're also seeing pictures from when the HVTN held its international meeting here in Seattle. Our CAB hosted an event that was a sort of festive event for community members from around the globe who came here to Seattle. And the final picture in the corner is a CAB retreat where once a year the group gets together to sort of tackle some issues in greater depth and make plans for the subsequent year. And then I have a few more visuals because I wanted to show some of the things that our CAB does, but also our community outreach team, the community educators and recruiters who work in outreach, attend events, and collaborate with other groups in the community. So these were shared by James and some of our other CER members who I'm not seeing. Well, I'll just look out in the audience. A number of you are here. This is a picture from last year's Pride Parade, and again highlights the antibody-mediated prevention study that we've been talking about happening in the Americas and in sub-Saharan Africa, but also our local collaboration with Wes Hurley's Capitol Hill series, which is a series of online videos that has also highlighted our research. So I thought it showed both things together nicely. This is from a community forum with one of our community icons leading a panel discussion, again with members of our outreach team. And then here's an example of outreach in action. So members of our outreach team will go out and put tables out any place where they can. So you may see them out on Broadway, out at the farmer's market, outside a bar, outside the library, any place where there is visibility and where they can talk with people. They'll be talking about HIV vaccine research and study participation. This is another picture from a Pride Parade a few years ago where we walked in collaboration with the Hutch Diversity Council. Members of our outreach team, this is at Lifelong AIDS Alliance and AIDS Walk, presence there. And this was another collaborative event. This was at Legendary Children at the Seattle Art Museum, which was again a collaboration between a number of different groups here in Seattle. So that's what outreach looks like in Seattle. And then we get to recruitment. And so I have the same information that Gail shared, that we are, again, looking for people to enroll in our studies who are between 18 and 50, HIV negative, and understand the potential risks and benefits of participation. So our outreach team also moves from outreach and education to recruitment. And this is a cartoon actually showing members of our outreach team about to work on recruitment. And we do use all of the modalities that Jim talked about being used around the world as well. A lot of community outreach, definitely having a presence on the internet, Facebook, and other social media sites. We also advertise on Craigslist and other internet classified ads. And we also rely in large part from referrals. And unlike research where referrals are coming from somebody's doctor, our referrals are coming from our participants in our studies who are referring their friends, acquaintances, family members to also participate in studies. And I'll show you the numbers on that in a little bit. But beforehand, here's just, again, some examples of the ways in which we do recruitment and advertising. This slide and the next one are two ads that were put together for us by a firm that did this work pro bono. The figure there is supposed to be HIV getting tackled, and had our information for contact on the side there. And then I also really like this one. This is from quite a number of years ago, but I wanted to include it. This was taken at Red Square at University of Washington, and the hammer is made up of our clinic staff members, CAB members, and study participants who all gathered to create this ad that, again, would have our contact information and let people get in touch with us if they're interested in participating in a study. And then we were talking earlier about community participatory involvement in education and recruitment. So this is a few ways that how this plays out in Seattle with the Sisters of Perpetual Indulgence, and helping to provide informational material about a vaccine study. This one started about 8 years or so ago. They've continued to work with us. And then this is a more recent example. Again, the antibody-mediated prevention study that we've been talking about is ongoing right now. We will include information about the study with condoms and packets of information with the condom, and provide study information about the study itself, as well as contacting us. And the trading cards are pictures of different community icons, and I'm not sure how many there are, but there are quite a number that represent members of the community who have agreed to be involved in, again, promoting our research. So what does this look like from our perspective of then being in the clinic, as I usually am, and how does somebody come from being in the community to coming to see us in the clinic? So our outreach team is collecting names of people through community outreach. We also get phone calls, contacts over the internet, and then the person will undergo a pre-screening conversation over the phone with one of our outreach workers, where they hear the general information about the study and will be asked a number of questions about their medical and sexual history that would enable the person talking to them on the phone to determine whether they are interested and potentially eligible to join one of our currently enrolling studies. And once someone appears eligible from the phone pre-screening, they're scheduled to come in and see me or one of our other 4 clinical staff for an in-person screening appointment, where we will talk about the currently enrolling studies in greater detail, obtain informed consent if somebody's interested in participating, get their medical history, their lab screening tests, and look at having them participate in the study. As we do all this, we're definitely tracking our recruitment sources, and I'll again show you our numbers in just a moment. But to put those numbers in context, I guess I would start by saying that we have enrolled nearly 2,000 people in vaccine studies and other HIV prevention studies since our unit started nearly 30 years ago. So I don't want to show you 30 years of data, so I just have the past 2 years. And so these are the data from beginning of 2016 to current. So just, I guess, about a year and a quarter, and the numbers vanished. So I can, not sure where they are, but I can tell you what they are. They were there earlier today. So we've had 1,811 people contact us in just over a year. And the, see how I can point to this, the light gray sort of largest piece of the pie is the 700, and I can't remember the exact number, but 700 some odd contacts of people who came to us by outreach. So that was our largest number of people, one-on-one, somebody talking to somebody in the street, somebody meeting somebody at an event, the personal connection, and then getting in touch with us. The next largest piece of the pie in the darker gray is the internet, and that I think was 671 contacts. And so I guess I like to think of this as, wow, the internet has this huge reach, but we actually get more people coming in to see us for studies from personal one-on-one contact than the internet. And then the next largest piece of the pie, the sort of teal or turquoise one, is radio advertising. We have advertisements on C89 and Pandora. People will hear the radio ad and call us up, and we always try to get somebody on the phone with them right away, trying not to have people go to voicemail, although we try not to answer the pager at 2 a.m. for those calls. And then the next largest one that is the darkest triangle toward the top is, I think it was 76, but it might have been 96. It's in that range, number of people who came to us from referrals from other study participants. And so I really look at this as nearly half of all people are coming to us by direct person-to-person communication. Let's see if we have numbers on the next one. Yes. Okay. So here are what happens to the 1,811 people who initially contacted us. 241 of them were interested enough and appeared eligible enough by phone pre-screening to come in for an in-person screening visit. And nearly half of those people ended up enrolling in a vaccine or other biomedical prevention study. But you can see even with those numbers, and the ratio is over 17-to-1 contacts for each person who actually ends up getting enrolled. So Jim's numbers earlier about the number of people who the network is hoping to enroll across the country, I think this makes it even more sobering of how many contacts one might need to make to come up with those numbers of people. And the other thing I just wanted to quickly mention about our clinic is that we also conduct a number of observational studies where people who may either not be interested in joining a vaccine or other interventional study or not eligible for a vaccine study may join an observational study where they provide blood samples or some of the other biopsy samples that Maria was referring to help us further understand immunology and provide information that plays into HIV vaccine development. And so people are contributing even if they're joining a study that is not, where they are not directly receiving an experimental product. And so 53 people who joined observational studies over just the past year plus. And then I just thought I would spend one moment talking about why do people not enroll. So a lot of it is definitely eligibility criteria. Especially early phase studies have very strict criteria, not only for age, but also for lab results, for sexual history, and for medical history. So certainly a large number of people end up screening out, as it were, because of eligibility criteria. There are also people who clearly end up being not being interested, who are lost to follow-up after screening. And then probably the other most significant factor is that aspect unique to HIV vaccine studies is a phenomenon called VISP, or vaccine-induced seropositivity. And this is referring to the situation in which many participants who receive an experimental HIV vaccine will have positive antibody results from having received the vaccine. They do not have HIV infection. They'll have a positive HIV antibody test. So that has a lot of implications in terms of not being able to donate blood. How do you do HIV testing in the community? And so people work with us on those issues, but that's another reason that people decide not to enroll. That could be a talk in and of itself. So I'm happy to answer questions about it later, but we'll leave it at that for now. And then the other factors are things that I've listed here: time, confidentiality, location, concerns about the study procedures, as well as side effects of the study product, and family or friends who don't want people to volunteer. With all that, however, though, as I get to my acknowledgements, and I have a slide of our principal investigator, Julie McElrath, our clinic staff, and I've pointed out our CAB and community partners, I definitely do mean the capital letters and the underlining for the study participants, because the reason that most people tell us they do enroll is altruism. And so seeing all these people who do enroll in studies to help with these efforts, and that their main factor is altruism, I want to end by pointing that out, and not quite end because I have one more slide, to say look for us in 2017. These are pictures from Pride and End AIDS Walk in the past. We'll clearly be at those events and many more over the course of the next year. Look for us out in the community. Thank you very much.
I don't know how we can possibly thank all of these people for the work that they do, but let's give them a round of applause and then we'll transition. What I'd like to do to respect your time and also give those of you who'd like to interact and do some Q&A, let's do a quick stand up and stretch. If you want to stay for the Q&A, you can sit where you are. You can move down a little closer. Those of you that have to depart, thank you so much for coming. And again, those that are willing to stay, we've got eager and willing Q&A about to happen. Let's give it about 30 seconds. Thanks.
Do we have our first question?
Okay, yes, I was just getting a little confused in this. When you're recruiting volunteers, it's in some of the things that were said, it said men and transgender people. What about women? Are women recruited also? Because that wasn't clear to me. And what kind of response? What proportions are you looking for? Or?
Every trial recruits a different population. So among the efficacy trials that I think were on Jim's slides, there were 4 currently in progress. 702 is recruiting both men and women in sub-Saharan Africa. The 2 sister AMP studies, the one in the US is recruiting men and transgender individuals. The one in Africa is recruiting women. And then 705 that'll be coming up in the fall is women in Africa as well. And then those are just our efficacy trials. So we're also doing early stage studies in men, women, transgender people, and each study is unique. The population is specific to that trial and the particular research questions that are being addressed. So it varies quite a bit. Our 2 AMP studies are unique right now in that the study in the Americas, this is the first HIV biomedical prevention trial of any prevention modality that has ever designated transgender men as an eligible population. So we're really proud of that. It's the first opportunity we've had to outreach to that community. We've had a few trials that have been specifically eligible for transgender women, but that's a whole new area of research as well, looking at what impact hormone use, for example, might have on one's vaccine responses and so on. So it's a whole nother set of research questions that we can address.
I have a question about community engagement. With so many research sites all across the world, how do you help the sites maintain a significant level of community engagement?
I just sick Gail on them. No, actually there's 4 Gails, and 2 are in Africa in Johannesburg, and 2 are here in the U.S. And I know Stefan was here before. He was the other U.S. guy, and I think he has left already. So they work very closely with them, and actually quite often Gail and the other 3 are simply on the road. They're traveling to all the sites. They're helping them, nurturing them. They help them make out a yearly plan. And in the plan, they really push them to make sure they're reaching out to the communities they need to connect with, not the ones that are just easy to connect with.
And Lewis is being modest because he was one of our community educators in Harlem up until about 6 weeks ago, and then we kidnapped him to come work at the Hutch.
So how were the sites selected? Were they random?
So it's actually a competitive process that we don't manage. We are funded through the National Institute of Allergy and Infectious Disease, and specifically their Division of AIDS. And the Division of AIDS has a grant funding process every 7 years. We go through one grant cycle and apply to be the network coordinating center, and the sites similarly apply to participate in the various research networks, and they have to present their capacity to do the trials and indicate what kinds of trials they're interested in doing. So do they have the capacity and interest to do vaccines, microbicides, treatment, work with mothers and children, and so on? And so they have to sort of make the case about what capacity they have. And that is a competitive process. It undergoes pretty significant peer review. I had the opportunity once to sit on one of those peer review panels. It's a slog, because those panels include, somebody who's an accountant and somebody who's a scientist and somebody who's an investigator, and the accountant is going, wait a minute, they think they're going to have an investigator for 40% of their time? But that never happens. And I'm like, no, that means that's a site that's going to be really successful. You have to give them the money for an investigator at 40% of the time. So there's this tug of war that goes on. So that we are currently on a cycle that runs through 2020. The next competitive process will probably get underway in 2018-19 for applications that'll be due and reviewed, with funding to begin in 2020 going forward the next 7 years. So we basically work with whatever sites come through that competitive process successfully and that are assigned to us by our colleagues at the NIH.
I think we'll take one question here, and then we'll get Bill over there, and then we can go into casual if you like. We'll close it after the next two questions.
Yeah, I'm just wondering, like, with there so many sites, so how do they collaborate? Do they follow like a standard rules or like?
So I guess I can talk about that from the site perspective. There's definitely coordination from the network here at the Hutch, but for any protocol that we're working on, there are conference calls where we meet with the other sites, and there will be discussions about operational issues. There are separate discussions about recruiting for the studies. And so the sites are directly talking to each other through calls and sharing their own experiences. And there are lots of different groups through which that happens, the clinicians, the community educators, et cetera. And then twice a year, the network holds international meetings where we try to send a couple people from each site and again meet our colleagues who are conducting the same work elsewhere.
And I would say, and one extra layer on top of that, most of our sites are like Seattle in that they are the only clinic in their community. But we do have a couple of cities where there are multiple clinics, New York City, Boston, Durban in South Africa, Johannesburg in South Africa. And so in those cities, there's, I would say, an additional level of collaboration, because those sites that are working in a common area usually meet together periodically to figure out how not to step on each other's toes. And, what kinds of events they'll work on together, what kind of events they'll divide up. And Louis could probably tell about this. Our clinic is going to cover the Pride Festival in Harlem, but this clinic is going to cover the Pride Festival in the Bronx. And this one's going to go to the one in Manhattan, so that we're not all doubling up and double, committing the resources and that sort of thing. So I would say that's one additional level of collaboration.
This is for the speaker who spoke about the number of participants needed for each study. How is that number determined, and what happens if you don't reach that goal?
It's determined by statisticians and the protocol team, and I'm sure it's a very complicated algorithm that I could not explain very easily. But they simply need to calculate. In an efficacy trial, you're comparing, unfortunately, infections. So it's almost like back engineering, figuring if we run the trial for X number of years and then powering the trial accordingly, that we'd need so many volunteers actually involved in the trial to follow the infection rates over those number of years. And I'm sorry, was there a second part of your question?
What happens if you don't? Do you want me to answer that? Yeah, so because the numbers of participants are determined scientifically, there's always a plan written into every protocol that says we need to assess if this protocol is not only being followed, but is enrolling people at a rate that makes it seem like it will be successful. Because if you're not going to have enough people enroll that you'll actually be able to answer the scientific question, then you're continuing to have people participate who won't be contributing to knowledge. So there's always a safety plan written in that's not only looking at the actual safety of the product, but also is the study enrolling in a way that it will be able to answer the question it was designed to answer. And different studies will have different ways of doing that, but it's usually at pre-specified times over the course of the study.
Thank you. I think we'll go into casual if anybody wants to stay. I think we've got a house ball timeline going on. You can leave it if you want to stay, you can stay. Thank you.
Thank you.